Sequencing quality is very important in everything we do in NGS. At the same time, there's other factors that people should really consider when thinking about the impact of sequencing quality in their application. Because in some cases, the nature of the sample, where the sample is coming from, how many copies of each DNA has to be sampled to get an accurate sequencing information, they all become equal, if not more important than the raw sequencing quality coming off an instrument. When people think about sequencing quality, they usually think about the sequencing metrics being generated on a sequencer or an instrument, which is definitely one of the most important parts of the sequencing quality for any analysis. And this has contribution from the chemistry, the instrumentation, the optics, and the analysis. There's other parts, though, of the workflow that really contribute to sequencing quality and sequencing quality in particular. And this is what I wanted to focus on today in this question. And namely, I want to think about the effect of the sample itself. So, for example, samples that are paraffin fixed or FFPE fixed, they will have a higher degradation of the genome that makes them less suitable for some application than for others. There are cases in which the kind of library prep being used is going to be gating what quality can be assessed in both ways. It could be creating some bias, if, for example, it includes a PCR reaction, or could actually enable a higher quality in the end of the analysis if it contains unique molecular identifiers, for example. But even beyond it, when we think, for example, at the analysis and the role that analysis plays in sequencing quality, there are some elements in just simply what reference has been used for the analysis, what is the information content in the genome that has been used to match the sequencing information against. All of these factors will play an important role in what the final quality of the information content from the sequencing run will be. Yes, it does, but only to a point. We know, for example, from a number of studies that beyond the Q-score of 35, roughly, there is very little practical usefulness for any germline application. That doesn't mean that high quality is not useful. There are some corner applications in which this will be beneficial, particularly in offsetting some of the coverage requirements for some application. That said, though, high quality is not the only way to achieve the information that people might want to gain from their analysis. There are other solutions, chiefly UMIs or universal molecular identifiers. They can leapfrog the quality content of a sequencing run to levels that are well beyond what any application might require. Q-scores are used to estimate the probability of a base call being correct. And these are achieved in an instrument and analysis specific manner, and they result from a calibration from a very large sample of known data. Generally speaking, base calls could be assigned a specific Q-score on a continuous scale, but there are practical limitations to this solution, particularly in the sense that it will present a very high data and computational overload. So what happens in practice is the base calls are bundled into ranges of base Q-scores, which are then reported as a group. And a good example of this is what happened with the latest release of the NovaSeq X software. In this case, it was done a very careful assessment of what the actual Q-scores of the base calls in a run was. The underlying quality of the base calls didn't change. What changed is how the software reports it. And now in the later version of the software, you will see that above 70 to 80% of the base calls are actually above Q40. Illumina has a very long history of pushing the boundaries of quality in sequencing, and I think throughout the 25 years of its existence, including Solexa time, there's been a continuous push of bringing sequencing quality higher standards. This, of course, has been achieved by working on the chemistry, and we've seen the example of XLEAP SBS being brought forward also to improve the quality of a sequencing run. But there's a lot beyond it. There's a lot of work going on a sequencer in improving the flow cell chemistry, improving the optics, improving the imaging and the raw data analysis that all contribute to improving data quality. There's even more to it. For example, if we think about the pre-sequencing steps, a library prep we know is key in determining data quality. And we're seeing a lot of improvements there in Illumina, thinking about more and more solutions that do not require a PCR step, so don't introduce a bias, as well as the introduction of the Illumina Complete long-read solution that achieves a different kind of quality by allowing longer range information to be acquired on the sequencers. Conversely, on the other range of the workflow spectrum, the recent innovations in Dragon and all the tertiary analysis solution that Illumina has been producing, they also push the quality to a higher level by really enabling customers to get the information they want at higher confidence. So data quality is a proxy for the quality and the performance of the sequencer. So the straight answer is yes. A good quality, a high quality means the system is working incredibly well and there's a high performance. And this goes to the point that the Illumina has invested decades of work in improving this property. The quality of Illumina sequencers, the robustness, the stability, the performance of the reagent, they all aim at providing the better answer for our customer at the highest confidence, which basically means the sequencing quality has been kept high and improved upon platform after platform. It's also worth noting that sequencing quality is not everything for an analysis. It is so very important for a customer to think about the application they're working on and how quality influences the information content they desire and they're hoping to get out of that application. In some cases, of sequencing instrument, like the raw quality of the sample, for example, might be more rate limiting. In other cases, the ability to sample the inadequate number of molecules could be more critical for the analysis than an excessively high sequencing quality. So quality is absolutely key, is very important, is one of the elements has to be considered when thinking about extracting information from a sequencing run.